This is part 4 of the lecture on chapter 26, and we're beginning this portion by looking at the molecular biology approach to making determinations of relatedness evolutionarily. So by comparing nucleic acids or other molecules, relatedness can be determined, and it's a valuable approach for tracing evolutionary history. And this practice has greatly accelerated the revision of taxonomic trees. Because if homology can reflect common ancestry, then comparing genes and proteins among different organisms should be able to provide insights into the evolutionary relationships that might not be available in other ways. For example, if two species have very recently branched from a common ancestor, their DNA is going to be much more similar, and their amino acid sequences should also be more similar. So, for example, if we have, again, groups that are so closely related to each other that they have not yet had a chance to have very much morphologic difference in evidence, by looking at their DNA, we can find more minute differences and minute divergence. And at the other extreme, some groups, for example, fungi or animals and plants, have diverged so much that they have little morphological homology left. Yet if you look at their nucleotide sequences, you may still find some common ancestry. So listed on this slide are different ways that molecular data is used to compare different species. In some regions, actually the rate of change in DNA sequences really varies from one part of the genome to another, and that's a very useful fact. So for example, with ribosomal RNA, there's a very slow change that occurs. And that makes it useful when you're investigating relationships between taxa that diverged hundreds of millions of years ago. But if you want to look at something that has more recently evolved, or if you want to look at species that are more closely related, or even populations of the same species, then it's helpful to look at mitochondrial DNA, because that evolves very quickly and so is able to show some of the differences in the more closely related or more recently related species and organisms. The first step in DNA comparisons is to line up homologous DNA sequences for the species that we're comparing. For two closely related species can differ only in which base is present at a few sites, where less closely related species may not only differ in bases at many sites, but there may be insertions and deletions that alter the length of the genes. As you were seeing as you did your BLAST lab, you could see how there can be small differences and vast differences between the organisms that you took a look at. So again, the first step is to align the homologous DNA sequences and try to find where there might be deletions or insertions. This is looking at the differences within the bear family here. This is going to step through how a computer program or mathematical tool might be used to compare DNA segments from different organisms. So here we've got the homologous stretch of DNA lined up, and then what we're going to do is, advance the slides, is see where there's been a deletion or an insertion. So here we show where that deletion has occurred between the A and the G on the first stretch of DNA, and then where the insertion occurred of GTA between the T and the C. So what we end up with is this bottom segment here, where we end up with spaces where things do not line up any longer, whether either from a deletion or an insertion. And as you saw with the BLAST program, you did see those stretches of dotted lines that showed where there were mismatches or no longer lined up matches of nucleotides. So again, I want to emphasize the point that the different types of DNA or nucleotide material is helpful for different reasons. So the DNA that codes for ribosomal RNA changes very slowly and is useful when investigating branching points that were hundreds of millions of years ago, where mitochondrial DNA evolves very rapidly, and so that makes it useful to explore more recent evolutionary events. I want to just define these two terms for you. Orthologous genes are found in a single copy in the genome, homologous between the species, and they only diverge after speciation has occurred. The and I'm probably not pronouncing this right, but paralogous genes result from gene duplication, and they're found in more than one copy in the genome. So why is this important? We're going to take a look at that in just a few more slides. First, I want to take a look at this cladogram, which presents a chronological sequence of branching during the evolutionary history of these organisms. So a cladogram presents this information, and it does not indicate the time of origin of the species that we're comparing, but serves to show the groups to which these different organisms belong. For example, a particular species in an old group may have evolved more recently than a second species that belongs to a newer group. A clade is a group of species that includes the ancestral species and all its descendants. Cladistics groups the organisms by common descent, and their clades can be nested into larger clades, but not all groupings of organisms qualify as a clade. Another way that we define different types of these trees is to look at them as monophyletic, paraphyletic, or polyphyletic. So the first one, a monophyletic taxon, and remember a taxon is any group of organisms given a formal taxonomic name. So a monophyletic taxon is defined as one that includes the most recent common ancestor of a group of organisms and all of its descendants. Such groups are often also called holophyletic. So you can see with the purple picture right here, this is a monophyletic group here. This is a monophyletic taxa because it's showing a recent common ancestor and all the resulting ones. And oh boy, we don't want that right now. Go away. Okay, the next one, the paraphyletic, includes the most recent ancestor, but not all of its descendants. So it may just show this one line. If it was monophyletic,